Welcome students, this is the last lecture of carbohydrate series, and in this lecture we will discuss the heteropolysaccharide, which we call as peptidoglycan, and it has a very good significance so far as the cell wall of bacteria is concerned. Let's see what is the peptidoglycan, what is its structure, and how we can inhibit the formation of peptidoglycan by using some antibiotics. We can see all these things in this video. First of all, peptidoglycan is a structural polysaccharide. It is a structural polysaccharide. I told you we have two types of polysaccharides. One is the homopolysaccharide and second is the heteropolysaccharide. Homopolysaccharides we have discussed, and heteropolysaccharides, which are made up of more than one type of monomeric units. These heteropolysaccharides, we have so many examples, like we have hyaluronic acid, we have chondroitin sulfate, we have these which we call as chondroitin sulfate, hyaluronic acid. They are structural polysaccharides. Even the peptidoglycan is also the heteropolysaccharide. So this is a structural polysaccharide, but a heteropolysaccharide, right? And this peptidoglycan is made up of, being a heteropolysaccharide, it is made up of two monomeric units. These two monomeric units are, one is the N-acetylglucosamine, and second is N-acetylmuramic acid. We call this N-acetylglucosamine as NAG, N-acetylglucosamine, and N-acetylmuramic acid we call it as NAM, NAG and NAM. In simple words, we say it NAG and we call it as NAM, N-acetylmuramic acid and N-acetylglucosamine. These are the monomeric units. If we see the structure of N-acetylglucosamine, both the two, whether NAG or NAM, are in beta form. That means OH is on the lower side. If we have to draw the structure of NAG, N-acetylglucosamine, we have to make a glucose, just like this, a glucose. And here we have a glucose, but beta OH should be on this side. And at position number 2, we have N-acetyl. It means it is NH and acetyl group, COCH3 group here. This is N-acetyl. Then you have OH, and CH2OH. This is your N-acetylglucosamine, right? NAG. And when we want to draw the N-acetylmuramic acid, we have to make again N-acetylglucosamine. We will again draw the same structure, that's N-acetylglucosamine, this way. Here we have again, it is beta, OH should be on this side, here it should be H, and it is again N-acetyl, just like N-acetyl we have to draw it. It is NHCOCH3 will be there, no difference. Here you leave this position, here we have OH and it is also OH, and here we have CH2OH and it is also CH2OH. Here at carbon number 4, at carbon number 3, this is 1, 2 and 3, 4 and 5 and 6. At carbon number 3, we have a lactic acid group attached to it. For example, I will tell you this is O. With this O, there is a lactic acid group attached. It is CH2. With this O, there is a lactic acid group attached. It is CH2CHO. Okay, CH2CHO and then C double bond O. This is lactic acid attached to it. And in this N-acetylmuramic acid, there are 4 peptides. There is a small peptide chain. Peptide chain means it is made up of amino acid, a small chain of amino acids. The total number of amino acids in that small chain is 4. We have 4 amino acids, and these 4 amino acids are here. Like we have alanine, ALA, we have alanine, we have this I can say glutamate, GLU, isoglutamate, we have lysine, and we have alanine back. We have a small stretch of peptide chain made up of alanine, isoglutamate, lysine, and alanine. Remember, all the amino acids are basically stable in L form. We have mostly, the proteins are mainly formed of L amino acids. So this is L-alanine, this is L-isoglutamate, this is L-lysine. But the alanine, the last alanine is not L, it is D, it is dextroalanine. It is D-alanine. Somehow, this is the structure of N-acetylmuramic acid. We need to understand because they are the building blocks of a polypeptide chain. So, here we have N-acetylglucosamine, very simple structure. N-acetylmuramic acid. In the muramic acid, we have an acetyl group at position number 2, same as that of N-acetylglucosamine, but at position number 3, we have a lactic acid group attached, and this lactic acid group is attached to a short polypeptide chain which is made up of alanine, isoglutamate, lysine, and alanine. But the first alanine is L, and the last alanine is exceptionally in D form. So this makes the structure of these two. Now, what is then peptidoglycan? Peptidoglycan is made up of these two things. One is N-acetylglucosamine, N-acetylmuramic acid, N-acetylglucosamine, N-acetylmuramic acid, and so on. It will be simply the combination of all those monosaccharide units, which we have discussed here. That is N-acetylglucosamine and N-acetylmuramic acid. Let us see, in this structure, we have some peculiar things which we need to understand. And what are these peculiar things? I will show you the structure. It is a 3D mesh-like structure. Peptidoglycan. And this peptidoglycan, you will find, is made up of 2 monomeric units. One is the N-acetyl. Okay, this is N-acetylglucosamine, N-acetylmuramic acid. Let it be N-acetylmuramic acid. And here we have N-acetylglucosamine. Similarly, we'll be having N-acetylmuramic acid, and this is N-acetylglucosamine. This is N-acetylmuramic acid, and again N-acetylglucosamine. Repeating units. We are getting the repeating units. This is a polypeptide chain. Right. Here also we'll be having the same thing. Let me write a little down. Alright. We have this and we have this. We try this way. This is one more. One more chain we'll be having here. For example, 1 and 2. Repeating units are repeating continuously. N-acetylmuramic acid, N-acetylglucosamine, N-acetylmuramic acid in black, and N-acetylglucosamine in this green. Again, we have so many other units like N-acetylglucosamine, N-acetylmuramic acid, black, N-acetylglucosamine, N-acetylmuramic acid, and N-acetylglucosamine. Right? This is the peptidoglycan chain. And in this peptidoglycan chain, you will find with this N-acetylmuramic acid, you have find attached to this N-acetylmuramic acid what? A short stretch of polypeptide chains. A very small stretch of peptide chains made up of 4 peptides. Let me try it with red. 4 peptides. This is 1, 2, 3, and 4. Right? Again, with N-acetylmuramic acid, you have find attached at position number 3 with the lactic acid a small stretch of polypeptide chain made up of 4 peptides. Again here you will be having the same polypeptide chain 1, 2, 3 and 4, and here also you will be having attaching to N-acetylmuramic acid, a part of N-acetylmuramic acid, our small polypeptide chain, and I'm drawing it everywhere, showing it everywhere. This is the polypeptide chain. This is also a short stretch of polypeptide chain associated to N-acetylglucosamine. Right? Now, what is there? There is we can say cross-linking. We can say there is cross-linking. There is formation of a pentaglycin interbridge. In this structure, there is formation of pentaglycin interbridge. How this pentaglycinic interbridge is formed? You will see the pentaglycinic interbridge will be formed by the combination of, by joining of third peptide, third amino acid of one peptidoglycan chain with the fourth amino acid of next peptidoglycan chain. For example, see, this is a structure, but this peptidoglycan structure is not very stable. How to make it stable? How to make it more stable? There is formation of pentaglycinic interbridge in such a way that the third amino acid of one polypeptide chain will be binding with the fourth amino acid of second polypeptide chain. See, this is the third. This is the third one. This will be binding with the fourth one. This is fourth. But it is a pentaglycinic. There are 5 glycine. 1, 2, 3, 4, and 5. There are 5 glycines. Again, the third with the fourth. 1, 2, 3, 4, and 5. Pentaglycin. Here also you will find third with the fourth. Third. 1, 2, 3, 4, and 5. This is the pentaglycinic interbridge. Again here third with the fourth. 1, 2, 3, 4. This is 4. Only 4 we have to try. 1, 2, 3, and 4 and 5. Pentaglycinic bridge. So this is a pentaglycinic bridge, which is making the cross-linking in peptidoglycan, and this making the structure of peptidoglycan a stable entity. Now, peptidoglycan has a very good importance because this peptidoglycan is forming the cell wall of both gram-positive and gram-negative bacteria, or we can say it is making the cell wall of bacteria. Bacteria follow the synthesis of the cell wall of bacteria. We require this peptidoglycan. And in the peptidoglycan, we have the repeating units of N-acetylglucosamine, N-acetylmuramic acid, and there is formation of a pentaglycin interbridge, and this interbridge is making the structure very stable. But remember, for formation of this interbridge, pentaglycinic interbridge, we require one enzyme, and that enzyme is known as penicillin binding protein. The enzyme is termed as penicillin binding protein or transpeptidase. This is the enzyme. For making the cross-linking of the two peptidoglycan chains, the cross-linking is provided by the pentaglycin interbridges, and for this activity we require one enzyme known as penicillin binding protein or transpeptidase. This way the bacteria is forming the cell wall, and cell wall is a stable structure of bacteria. Now we have seen in case of bacterial infections to which we are commonly the hosts of the bacteria, and we encounter a number of bacterial infections in our lifetime, and every time whenever we have a bacterial infection, we get relieved of by using certain drugs which we call as antibiotics. A special type of antibiotic, or we can say the most popular antibiotic, is known as penicillin. We give penicillin to get rid of bacterial bacteria. Penicillin is antibacterial. We are giving penicillin to get rid of the bacteria. And this penicillin, which was the first antibiotic, has the mechanism of action. When we see the mechanism of action of penicillin, it will bind on it will inhibit the penicillin binding protein or transpeptidase. It is going to inhibit the enzyme. When we give penicillin to any patient having the bacterial infection, we see the penicillin, the mechanism of action of penicillin is to bind with the penicillin binding protein or transpeptidase, and by this action, when there will be no penicillin binding protein, when there will be no transpeptidase, what will happen? The pentaglycinic interbridge will not be formed, and the stability of peptidoglycan is lost, and the cell wall of bacterial cell will get damaged. It will not be formed. It will not be formed at all, and we will get rid of this bacteria. That's why it's very, very important. This is what we call as peptidoglycan. Thank you very much for watching. Wish you all the best.